Thank you both for those presentations. And because the material is a little more diverse, I'm going to do this a little bit differently than the first panel. And I have a couple of questions for Dr. Kurian first. So we heard from that first panel about patient-reported outcomes, particularly integrated into care and integrated into self-care. And I'm curious if you have looked at the role of any digital biomarkers or patient-reported or patient-generated data in your model development, and as well as, and this was a question from one of the other organizers, as well as genomics. So just a general question about those other forms of data in your models, and then I'll give you my second question as well, which is, you talk about identifying, one of your first examples was identifying variation and anomalous variation. And I'm curious whether overall in the use of these predictive or prescriptive algorithms, you're concerned about reducing personalization and exploration that happens in the context of care. So I'll pass it over to you.
Well, excellent questions. And thank you again to the organizers for letting me talk about these things today. So we are very interested in including patient-reported outcomes. You know, at first, most of our sources have been things that are collected clinically, including in the EMR. At Stanford and Sutter, there have been moves in recent years, of course, to have a patient portal and routinely to collect patient-reported outcomes. And we are integrating those into this OncoShare data set. We've had one paper looking at sort of initial description of some of the patient-reported outcomes and are working on more in terms of delving into details about what patients say, and even data like how often are they messaging doctors and things like that. So that is being integrated, and thank you. The genomics question is tricky. And again, that is a challenge of really lack of integrated data into the EMR. As many of us know, often these are send-out laboratory tests that come back as a PDF, making them difficult to extract in terms of getting things into the EMR. I think this probably reflects the broader challenge of integrating genetics and genomics into the practice of oncology more broadly, right, and not having it siloed off to the side, both virtually in our data sets and in our actual practice. So I think that's a move that we need, and we're not quite there yet. I have dealt directly with the laboratories who produce the results to get the data, but I think we need to do better there. And then there was the question about variation. Yeah, I think that's very important. I'm a practicing breast oncologist, and certainly no one size fits all, and it's very important to get at these issues. I think one thing that was striking to us, again, in the example that I showed you, was that we really did have metrics of disease severity, including genomic scores like the 21 gene recurrence score, showing that they were exactly the same in terms of all prognostic factors, and yet getting more invasive types of every intervention we found. And it's not that that would always be wrong, but it certainly creates a trend that we think warrants investigation. And this kind of triangulated data source allows us to recognize that.
Thank you so much. So now a couple of questions for you, Dr. Parikh. Fascinating work. And my questions are, first, what is sort of, if you will, the mechanism that you think is really at play? Is it about the importance of identifying those patients that you point out? Is it making the clinician more comfortable, or sort of, as you referred to in that spillover effect, is sort of success of all of this that ultimately you retrain clinicians to have an overall a tendency to do this more, and then they don't really, in some sense, need that particular tool. And so maybe some of these prescriptive algorithms are as much about training the clinician as it is in an ongoing tool. And then my second question was, I'm sure you've been watching everything that's going on in terms of end of life and serious things in the context of COVID. And there's both a learning thing from your tool to there, but also perhaps the unfortunate availability of so many of those conversations and how they clinicians take them on there. And so I was just curious if that's something you thought about. And then after you answer, I can bring in some other questions.
Sure, those are two great questions. I would say when it comes to the mechanism at play, when we showed, and this was in work that we did early on when we displayed predictions to clinicians, I would say that for the vast majority of patients, no one was surprised that an algorithm identified those patients. I would say there was very few surprises there. And I think we regarded that as a positive thing. I think that if you've got a patient in front of you, an early stage breast cancer patient that's likely to survive past 6 months and it maybe is being flagged because of adverse laboratories or something like that, a clinician may have distrust in the overall algorithm process in general and not even proceed towards acting on the intervention that you have tied to it. So I think that more so, I think that the money in this type of intervention is tied to the automated identification. One of the biggest barriers towards identifying patients who are appropriate to serious illness communication is time. And the fact that if you put a list of patients at the beginning of a busy clinician's day, chances are they'll identify tons of them that are appropriate for the conversation, just like everyone should be appropriate for the conversation. The issue is, who do I have the conversation with today? And I think that in integrating that process into the workflow, and it wasn't it was as seamless as I would say you could get in terms of integrating this, one text message a day for patients who are up to 6 patients a week who are identified as high risk. We strove not to make this burdensome on clinicians so that we could actually try to change behavior. I think the automation and the integration into routine text messages was sort of where I think the secret sauce of this type of intervention, and really, illustrates that it's not just the algorithm that's going to positively change behavior, it's the intervention that you tie it to. I think for the point of COVID, I think it's been really interesting because we deployed the intervention over a year ago. So at the time that coronavirus sort of started to hit the Philadelphia area, we were really, it was baked into pretty much all of our tertiary medical oncology practices. And we still saw really significant, well over 10%, well over 12% for high-risk patients, documentation of serious illness communications for patients deemed at high risk, sort of sustained levels, even during the pandemic. Interestingly, much of these happening over telehealth, which we have discrete codes to identify. So I would say the process of integrating the algorithm into the workflow really had some positive effects beyond just the overall trial period.
Thank you very much. I'm just looking if there are any other unaddressed questions on the chat. I'm not seeing any. I wonder if you have any last comments or things you'd like to say in our remaining minute and a half. Either.
Sure, I can start. I think that the integration of data, across multiple different disciplines and different sources, and the ability to demonstrate performance metrics that we've never seen before with traditional algorithms, and whether they be diagnostic or predictive, I think is really, really key here. I think that, over the next 5 to 10 years, the key is going to be getting these into actual clinicians' hands and testing them prospectively in rigorous fashion, not just pre-post analyses, but actual rigorous testing like we do diagnostics and drugs to make sure that this money is worth the investment. And I think if we do that, we'll be able to make much more inroads in applying predictive and diagnostic analytics in the practice than we have so far.
And I agree completely. I would just comment, I think that there's a need certainly for scaling, right? I'm giving you a demonstration from 2 healthcare systems, but scaling bigger. There's certainly work in the SEER program to begin to integrate some of these data sources, which I think will be helpful. There, I think, is also need for a real-time integration, right? Because SEER often has a lag in terms of case collection. So those, to me, would be key priorities here.
Thank you both so much, and thank you to all the speakers in this session. And I believe that Larry wanted to talk about some logistics.
Yeah, thank you very much, Deborah and Randy, for your moderations and for all the speakers. I think these were incredibly salient examples of what's actually going on now and the possibilities that we have. So I think it was a terrific way to sort of launch the rest of the workshop. We're going to take a lunch break. I apologize for not being able to feed you lunch. If we were meeting in person, we would. But we're going to take a break between now and 1 p.m. We will start promptly at 1, and our first session is going to be a conversation between myself and Amy Abernethy from the FDA, which I think is going to be a critical one for us to understand how the FDA is viewing digital technology these days, before the COVID pandemic, and how the COVID pandemic has changed their view of things, if it has. So have a good lunch, everyone, and we will see you back at 1 o'clock. Thank you all.